Hello, people. I hope you can all hear me, see me. I'd appreciate someone confirming just in the chat so I can actually get started. We're just going to wait a couple of seconds, so maybe other people that want to join can still join, and then we're going to get started. Well, okay. I'm going to start introducing myself again. We can wait for a few more people to join. But I think we should get started right away. So I welcome you all to yet another lecture of physiology. I'm glad for everybody that came. And today we're going to be discussing the cellular basis of the nervous system. It is a slightly more complex topic than last week's, and I'm going to try to focus on the aspects that are more difficult to explain rather than the aspects that you just need to memorize. There are certain aspects you need to memorize, there are certain aspects you'll have to understand in order to be able to recite them in the actual final exam. So I'm going to point out at the individual spots where I think that is the case. And again, just like last week, in case anything is confusing, anything I haven't made clear, or if I'm talking too fast, or any other questions, please just drop me a line in the chat and I will try to get back to you as quickly as I can. And at the end of the lecture, I have done a test run and it shouldn't be taking too long. So at the end of the lecture, we're going to have plenty of time to discuss other questions if you feel like it, if you've got anything on your mind. So we're going to be discussing, first of all, the structure, like the cellular structure of the nervous system, which is basically neurons and their supporting cells. Then we're going to discuss nerve signals and their generation. That is probably the core of this lecture because it is the most complex one to understand, and we're going to focus a lot on that. And then also the working mechanisms of synapses. And last, neurotransmitters. So without further ado, we're going to get started with neurons. The structure of a neuron should be reasonably clear to most of you. You've all done biology. So the basic structure is like any other cell, pericardium filled with endoplasmatic reticular, Golgi, nuclei. And the pericardium is their basic function is for neuronal housekeeping. So a cell is like a neuron is a living thing. It has to maintain proteins, it has to produce cell membrane proteins, etc. So all of that happens in pericardium. We have dendrites, which are the receiving end of the cable. And we have an axon, which is the outgoing end of our quote-unquote cable. Dendrites act as a passive cable. That term, what that means, I will come back to later when we're actually discussing the physiology of the neuron. They're responsible for information retrieval. The axon is an active cable. It starts at the axon hillock. Right, whoops, that was a mistake, I'm sorry. Nope. Go back, thank you. The axon hillock right here, and it is active cable all the way down to the synaptic terminals. The axon itself features microtubule, neurofilaments, and a very sophisticated transport system that we should look at in detail because it should definitely come up on your partial tests and if not on your final exam. The transport system, and I'm going to stop this animation because it's actually quite annoying, but it's very nice to illustrate. You have the cytoplasm of the actual axon is filled with small microtubules. If you remember from biology, the microtubules have polar end. That means the one end is positive, the other end is negative, so to speak, based on the structure of the microtubule. You remember that they're like basically small tubulin proteins attached to one another. And based on that polarity, there's two main types of molecules that actually allow for the transport of, for example, vesicles down or up that tubule. For the fast transport system, we have kinesin, which allows for the transport of vesicles downstream, so anterograde. And we have brain dynein, or MAP1C, which is responsible for the transport retrograde, or towards the pericardium. Why do we transport things along the cell body? Well, we need to maintain membranes. We need to maintain the membrane-bound proteins. That includes channels, that includes the receptors. That kind of stuff will be transported within vesicles for the most part. And these transport systems are energy dependent. The fast transport systems have a higher energy demand than the slow ones. The slow ones are mostly responsible for cytoskeletal components. What do we need to transport back to the pericardium? Well, the cell will actually take up a lot of extracellular components that it comes in contact with, for example, toxins, growth factors, or viruses, and those will be transported back in a similar manner as to this walking animation displayed here, back towards the pericardium. Next, we have the basic classifications of neurons. This is, again, something that I'm afraid you will have to memorize. There's not a lot of explaining that I can do here. First of all, I'm not quite sure if you've actually covered this in histology just yet, so I'm going to discuss it a little bit. The external projection would be the first type to classify a neuron. We have projection neurons that basically project from their site of origin to a completely different site within their brain, for example, within the brain or within the brain stem, spinal cord, whatever. So, for example, having a neuron that projects from the cerebellum to the cerebral hemisphere. That would be a projection neuron. And the other class would be interneurons, which are small connective neurons within their individual system. For a histology exam, you will be able to have to draw this small outline of the cerebellum, of the cerebral layers, and also of the cerebral cortex, the individual layers. For the physiology exam, you're actually going to have to draw the layers and their small connective networks, at least for the cerebellum, if I remember correctly. But you're going to learn that in neurophysiology in the future. That is sufficient for now. Then the other significant factor would be dendritic geometry. It can either be pyramidal or stellate, as, for example, in the example 3 down here, that would be an example of a stellate dendroid. Example 2, this one's up here, would be an example of pyramidal. And last but not least, but for us in physiology most relevant probably, is the number of processes. We have unipolar, which is the example in number 1. Bipolar, which is one dendrite and one axon. So this part is the dendritic section, this is the axon section. Multipolar, where we have multiple dendrites arising from the pericardium and one single axon leaving. And then we have pseudo-unipolar, where the pericardium is located halfway through the axon itself. These are, for example, very important if you're looking at the afferentations and efferentations of the spinal cord. Next up is the nerve classifications, yet another class of nerve classifications. Again, something you'll have to memorize, and it is difficult to explain this right now if you've not yet covered muscles and other receiving ends, for example, like muscle sensory organelles. So the individual functions of those nerve fibers is going to become more clearer once you've actually covered those. I'm going to come back to this slide later, though, if we do start discussing the conduction velocity of nerve fibers. For now, this is simply a table that is very helpful to memorize. If you want to have more detail on it, it is originally from boron. It is very helpful to go through that. But I'm going to come back to that later. Now we're going to start discussing the core of this lecture, which is the generation of nerve signals. Just to recap, from last week's lecture, well, 2 weeks ago, the resting membrane potential is established through the continuous outflow of potassium. So the continuous outflow of potassium provides a current, and that current over a concentration difference is basically equivalent to our electric circuit, so to speak. The exact voltage of this electrochemical gradient is determined by the Nernst equation. And if we drop in the numbers, we can actually see that it gets us roughly towards our resting membrane potential. So resting membrane potential in most cells is solely dependent on potassium, but it's not the only thing. The exact resting membrane potential is actually calculated by this formula, which is the conductivity for a given ion, signified by G over G, the total conductivity for all conductor species, and the equilibrium potential for the given ion, so for example, for potassium or for sodium. Now, if we're looking at the formula and if we're looking at what we're trying to do, what is an action potential? An action potential is the rapid depolarization of a membrane. Now, we can achieve this in two ways. We could depolarize the membrane by simply switching off all transporting proteins and allowing the concentrations on both sides of the membranes to equalize. However, that would be a dramatic waste of energy because we have expended significant quantities of ATP to establish this concentration gradient. So simply waiting for these two concentrations to equalize would not be a particularly good way to go. The other way would be to change the conductivity for a given ion. So at rest, this is the only thing that is truly membrane permeable. But during an action potential, what we do is we increase the permeability for sodium for a fraction of a second. And that in itself is enough to change the membrane voltage to our depolarized state. In reality, this is going to look like this. We have a threshold potential, so a small depolarization that is sufficient to trigger the voltage-gated sodium channels. Voltage-gated means after a certain voltage, in our scenario, minus 55 millivolts, these channels are going to open. And the moment sodium influx arises, the membrane will be depolarized, it'll rise, it'll spike to plus 40, and then because the sodium channels are only remaining open for a fraction of a second, actually only 1 or 2 milliseconds, the sodium channels are going to close. The permeability for sodium is then practically back to zero, and thus the membrane potential reverts back to the resting membrane potential. In addition to that, the change in membrane potential actually forces more potassium out of the cell because at a depolarized state, it is more favorable for potassium to leave the cell. So we have a small overshoot actually at the bottom here, where more potassium left the cell and the cell is momentarily hyperpolarized. The hyperpolarization and the closure of the sodium-potassium channels leads to a certain effect that is called the refractory period. The refractory period is the time that is required for, first of all, the sodium channels to recover from their opening. If you remember what a sodium channel looks like, it's like a hollow tube with two lids. And during the opening sequence, both of them are actually open. Once the opening sequence has, once opening has occurred, just after a split second, one of them is going to close. And it takes a little bit of time for the other one to close back up as well and the other one to open. And this way, this is just a thing that happens in a given amount of time after a given amount of time. And this prevents the sodium channels from being this prevents the sodium channel from being opened again. During this time, it is called the absolute refractory period, the sodium channel will be unable to open. This is the absolute refractory period. The relative refractory period is the period of time where the sodium channels can be excited again. However, as you can see here, the membrane potential is lower than the initial resting membrane potential, which means the stimulus required to rise above the threshold level, which is up here, is far greater than at normal resting membrane potential. To overcome this, we need a far larger stimulus in order to elicit another action potential. So this is the relative refraction period. Nerve fibers in general work on the basis of the all or nothing principle, which means either we have a stimulus that is sufficient enough to depolarize the membrane to reach above the threshold level or not. And if it's not sufficient to depolarize the membrane, those are called failed initiations. We have a small decrease in membrane voltage, but it is not sufficient to actually cause the depolarization, the opening of the sodium channels. So how does this propagate down the cell? Imagine we have sodium channels stacked side by side. The moment one sodium channel opens, this region, I'm going to go back to the diagram before, the moment sodium channels opens here, the region right next to it will be slightly more depolarized as well. And if that reaches above a threshold level, this sodium channel will open as well. So you have sodium channels after sodium channels opening. Then another part of this is, so if we have how are we going to convey information down the stream? So we have sorry, I need to start over. How do we convey information? Imagine, for example, visual information. There's a continuous flow of information that needs to be relayed up your brain. And because of the nature of the action potential, we actually cannot change how high the voltage is. That means information is not conveyed through the amplitude of the potential, but rather through the frequency. If we have the baroreceptor, for example, in the carotid body, if you have a high blood pressure, it starts sending a high frequency signal rather than the continuous slow signals that happen under low blood pressure. I hope that is reasonably clear. Again, if you have any questions, if I have not explained something properly, please stop me and I'll try to come back to it. Now, we need to go back to how this actually propagates down the cell and what effect this has on the conduction velocity. If we have an unmyelinated axon, as explained before, it will simply depolarize the most closest section of the membrane and trigger individual sodium channels one by one. Now, this is a very slow process. The circuit is basically ions flow in here that will flow towards the still polarized segment of the axon and will trigger depolarization of that segment. But the circuit will loop back on itself at the closest possible opportunity if the resistance is low enough, because the circuit is trying to be as short as possible, because the longer a circuit, the higher its innate resistance. However, there's one way to overcome this. If we add a myelin sheath is practically nothing else but a very large resistor. And if we have a resistor along the length of the axon, the shortest possible circuit increases in length downstream the axon. That means the next possible segment that can be depolarized is a lot further away than if we have an unmyelinated axon. This means that the action potential will actually jump from one gap to the next one through something called saltatory conduction. Through this mechanism, the conduction velocity increases manyfold. We're going to look back at the slide that I showed you on conduction velocity and diameter. It is going to become very apparent there. But basically, the local current is the current that is produced by the inflow of ions, and that will propagate downstream along the axon and also in reverse direction outside the axon in order to close the circuit. And it will always follow the path of least resistance. So either the shortest, or if the shortest part is blocked by a myelin sheath, it will go to the next gap junction and flow through there. The region underneath the myelin sheath actually has no sodium channels because sodium would not be able to leave here. So it has no choice but to jump from one node to the next. Now we need to talk about something that is the local response, because if we have membrane potentials that are not sufficient to either depolarize the membrane Or we have something called a passive cable, which, for example, in this scenario is dendrites. There are no sodium channels that could be opened up through the change in voltage. The local current will simply dissipate towards the pericardium and depolarize towards the pericardium, but with decrement. So here, where we have excitatory synapses, we're going to have a very high or closer to the threshold potential membrane voltage as towards the pericardium. This is also true in reverse. We can have inhibitory synapses, which hyperpolarize the cell. So if we look at the resting membrane potential, I'm sorry about my drawing, and imagine this being our threshold potential, we can either depolarize it towards our threshold potential, or we can hyperpolarize it away from our threshold potential. So the stimulus needed to get towards a depolarization is far greater. In this passive cable theory, the local current will simply spread towards the pericardium, and if the voltage of the pericardium drops sufficiently at the axon hillock, then voltage-gated channels can open and actually send out the signal down the axon. There are multiple effects to consider here, which are very important. It's called summation. We can either have temporal summation. Imagine we have a synapse that sends signals on a very regular basis. For example, here. Okay, imagine this synapse here would send signals on a very regular basis, and there would be excitatory signals. That means our signal would look something like this. It will increase a little bit, then next signal comes, it'll increase a little bit, and it'll increase ever closer towards the threshold potential, because it does not have sufficient time to actually decrease back to the normal level. If the other alternative would be spatial summation. If we have multiple excitatory neurons firing at once, this jump is going to be instantaneous, closer towards the threshold potential. I just saw that there was a question, and I'm just going to I think I'll change it to long. So the someone just posed the question whether the myelin sheath, if it's too long or too large, would not get lost along, if the signal would not get lost along the way. I'm quickly going to jump back because this is important to understand. You have to keep in mind that this nerve or this axon is nothing else but an electrical circuit, and we need to be able to close a circuit. That means if a depolarization occurred here, the circuit or the current has to flow one way or another, and if the way or the path of least resistance is way downstream the axon, it'll close the circuit like that. If the resistance is too high, then theoretically we might lose our action potential. However, in reality, I don't think this is a phenomenon that occurs. It would defy its purpose. So I don't think this is relevant for our purposes at the moment. The thing is, like, this is the path of least resistance. So there is no other alternative for the circuit to close. So this way, yes, it'll have decrement to some extent, but it would still be sufficient to at least depolarize the next gap within the myelin sheath. Decrement, actually coming back to this is what happens in the passive cable. We have no sodium channels here that can be triggered through a depolarization. So it'll simply be the local current that slowly dissipates through the dendrite and towards the pericardium. Okay. Now, conduction velocity. What is conduction velocity dependent on? If you imagine that our axon is nothing else but a cable or a tube where water flows through, the larger the diameter of the tube, the more current you can shove through that tube in a given instant of time. That means with increased diameter, the resistance of the axon is going to decrease, and thus the velocity will increase. The current will spread faster through the axon. However, this effect is only minuscule compared to the effect of myelization. This is illustrated very well at the axon of a squid, sea fish. So a squid has an axon of a diameter of 1,000 micrometers, so almost a millimeter, exactly a millimeter. And the conduction velocity of that axon is only 25 meters a second. The fastest axon in the human body reaches up to 120 meters per second. And this is achieved through myelization because the spread of the action potential through gaps in the myelin sheath occurs a lot faster than having to depolarize every individual segment of the membrane itself. So if we want to look at what increases our conduction velocity, point number one would be the axon diameter. This is for class A alpha fibers, very important. They're the largest diameter fibers in the body. And we have velocities of 80 to 120 meters a second. For A beta to B fibers, the diameter decreases continuously. And with the diameter, the conduction velocity will decrease as well. C fibers are a special case because C fibers are actually not myelinated. They're an evolutionary old system, and it was simply still kept within the body. It wasn't selected out. And their conduction velocity is actually at a rather slow-paced 0.5 meter to 2.5 meters a second. So this illustrates the effect of myelinization and axon diameter on the conduction velocity. All the important fibers that innervate, for example, muscles, muscle twitches, or rapid sense information go through type A fibers. Next up, we need to discuss synapses. So the problem of relaying signals from one cell to the next was actually relatively clear. That was already discovered in the 19th century when the basics of electrophysiology were discovered. And two main theories were proposed. It was either that there was an electrical connection between two cells, or based on the fact that if you dropped small amounts of adrenaline on certain nerve fibers, I think it was adrenaline, I'm not quite sure, close to nerve fibers, you could actually see potential rising. It was also suggested that it might be a chemical synapse as well. And turns out both principles were correct. In the human body, we have two main classes of synapses. We have the electrical synapse and we have the chemical synapse. The electrical synapse is two cells or two nerve fibers linked by gap junctions where the actual cytoplasm of the individual cells is linked together. And the current will simply spread directly from one cell to the next. The best example of this is the myocardium. The myocardium comprises muscle cells that are either contracting or conducting. But all of these muscle cells are linked together. They form one continuous syncytium, is what it's called, where if you excite just a small region of the heart through, for example, the AV node, the entire heart and the entire myocardium will depolarize because the action potential spreads through the gap junction linked cells. The advantage of this is that it forms a so-called reciprocal synapse, where the action potential can propagate in both directions, no matter which region was excited first. The chemical synapse is not able to do that. The chemical synapse has a receiving end where the signal arrives. It has the presynaptic terminal and the postsynaptic terminal. And because we only have vesicles of neurotransmitter over here, it's only a one-way street this can go. A chemical synapse has a gap of roughly 30 to 50 nanometers, far too large for any electrical current to pass through. However, if we fuse our vesicles with neurotransmitters in the synaptic cleft, they will diffuse across rapidly within 1 millisecond and reach the postsynaptic terminal. And what happens there, that is what we're going to discuss next, because this is one of the most fundamental concepts of synapses and nervous transmission in general. The function of the actual nerve signal is dependent on the receptors. I forgot it. We're going to come back to that. First of all, I'm just going to recap how the actual synaptic transmission works. Action potential arises at the presynaptic terminal, where calcium channels will open. Calcium, voltage gated, calcium will diffuse into the cell. Calcium is a second messenger, like, for example, CAMP, IP3, etc. And calcium will bind specific proteins on the vesicular membrane and will allow for the vesicle to fuse with the presynaptic terminal. Neurotransmitter will then diffuse along its concentration gradient through the synaptic cleft and bind receptors at the other end. Now, this is where it gets interesting because we can have various classes of receptors for the same neurotransmitter. The neurotransmitter binds the receptor. It'll either diffuse away again, or it'll be removed through enzymatic action. For example, if you have an acetylcholine receptor or an acetylcholine synapse, cholinesterase will remove acetylcholine, and the remnants will actually diffuse back towards the axon terminal, will be retaken up and actually be recycled to produce more acetylcholine. So now let's get to the effect of the actual synaptic transmission. The effect of the synaptic transmission is dependent on the receptor that is located at the other site. In general, we have two classes of receptors: ionotropic receptors and metabotropic receptors. Ionotropic receptors are the classical channel that we discussed in the previous lecture, where a neurotransmitter binds and the channel will open and allow for the diffusion of specific ions. This can be any ion. This can be a sodium channel, for example, in the example of a nicotinic acetylcholine receptor. But this can also be chloride channels, which would actually hyperpolarize the membrane. Acetylcholine receptors are the most common example. They're found in most peripheral nerves and at neuromuscular junctions. They are highly important. Metabotropic receptors work on the basis of G proteins. G proteins trigger intracellular signaling cascades, so they can elicit responses within the cell. They are highly important, for example, in the regulation of the enteric nervous system, of the adrenal gland, all those. The most prominent example that I could suggest would be the muscarinic acetylcholine receptor. All parasympathetic postganglionic effectors are actually provided by almost all, I think, except the svetolins, but I'm not quite sure, are actually provided by muscarinic acetylcholine receptors. And the effect of acetylcholine binding this muscarinic receptor is dependent on the class of the muscarinic receptor itself, because there are even more subclasses of muscarinic receptors. There's actually 5. And depending on which one is attached to the membrane, the response within the receiving cell will be different, which means the function of the synapse is dependent not on the neurotransmitter, but actually on the receptors that are present on the receiving end. And that'll elicit a different type of response. This effect becomes apparent if you look at neurotransmitters themselves, because neurotransmitters can have multiple functions at once. Neurotransmitters in general, just to recap, you're going to learn this in far more detail than you'd like in biochemistry, by the way. Can be amino acids, monoamides, catecholamines, and they're employed in chemical synapses. They can be either excitatory, so depolarizing the membrane, or inhibitory. Examples include glutamine, acetylcholine, histamine, etc. There's one example that I'd like to actually focus on, because dopamine can be both excitatory and inhibitory. This is due to the fact that there is, again, like in the example of acetylcholine, more than one dopamine receptor, which can have opposing effects. This is very important to remember. Neuromodulators are the next example of neurotransmitters. They're not actually eliciting a actual potential. Neuromodulators will change the behavior of the receiving cell to either become more excitable or less excitable. They so-called modulate the response to a stimulus. Examples include dopamine, serotonin, acetylcholine, histamine, and noradrenaline. If the adrenal gland produces noradrenaline, a lot of neurons will actually respond to that and increase in excitability. This is a neuromodulator effect. That means it will take a smaller stimulus to elicit the same response as if noradrenaline was not present. The last part would be neurohormones, which are, for example, the hypothalamic hormones. What makes neurohormones? Well, neurons that actually release a neurotransmitter into the bloodstream, it becomes then a neurohormone because they're transported by blood. Most prominent examples would be ADH and oxytocin. I think this is actually it for now. One thing that I did not mention in this lecture that you will also have to look at is the supporting cells of the central nervous system and the peripheral nervous system, i.e. glial cells. Again, this is something that you will have covered in biology, will cover again in histology, and the physiological function is not covered in detail in physiology, so I decided I was not going to go into detail in it in this lecture, because I think the most important parts to get across in this lecture is how action potentials arise, what the reason for the action potential is, and what the response to different neurotransmitters are, depending on the type of receptor that is employed on the membrane. I am aware that this is like a lot to go through, so in case I haven't done something in detail, you have the chance now to ask me any question you'd like, and I will try to answer as well as I can. So make use of the chat as you please. Is nitric oxide also an inhibitory neurotransmitter? Nitric oxide is a hormone in the first place. It is produced, for example, in the endothelium of blood vessels in order to modulate the response to vasodilation. Now, nitric oxide is a vasodilator primarily. If we have a sympathetic I'm not quite sure. Sympathetic response, I think, in the endothelium, nitric oxide will be produced and it will diffuse to the smooth muscle, but it is not actually a neurotransmitter because it does not flow through a synaptic cleft. So I don't think it would actually classify as one. Laura asked, does the inhibitory excitatory effect depend on the neurotransmitter or the receptor or both? Well, a receptor most of the time actually only responds to one neurotransmitter. That means if the receptor is either excitatory for this neurotransmitter, it'll depolarize the cell. If it's inhibitory to the neurotransmitter, it'll hyperpolarize the cell. Yeah. So yes, the effect technically depends on the neurotransmitter because some neurotransmitters have a generally more excitatory effect or inhibitory effect, but the primary effect, if you're looking at one synapse, depends on what is at the receiving end, at the receiving terminal, what type of receptor is employed there. From what book do I recommend studying? You might have noticed from the diagrams that I've been using, I mostly take my diagrams from Boren Bullpaps or Guyton's. Both of them are excellent books. If you have time to go through them, I recommend it, but I recommend it running towards your final because it is very time consuming. But those are probably the two favorite books I've used for studying so far. If you still have other exams to take, for example, anatomy or histology, I would rather take one of the shorter books, for example, I forgot the name. There's a couple of shorter books that are also recommended by the physio department, and they will be sufficient to at least get you through the first and second semester. For the final, it is for some subjects definitely not sufficient to just look at those. Last resource I'd recommend, there is a excellent neurophysiology website that goes through all of neurophysiology, and neither Boron nor Guyton is actually sufficient for neurophysiology. The lecturer that is doing neurophysiology lectures, he usually links that in his slides, and I would highly recommend accessing that. Have I answered the question about nitric oxide and about the inhibitory excitatory effect sufficiently? If not, please give me another nudge. Great. Welcome. Okay. Well, I'm guessing this is the end of this lecture. In case you have any questions, feel free to reach out. And I appreciate all of you for coming. And I wish you a very healthy, hopefully, and good coming few weeks. And I hope to see you again soon. So, bye-bye.